Hello, everybody, and welcome to the final of the three question times live from Boston, ACTRIMS-ECTRIMS 2014. So we're really glad you can join us today. We're looking forward to taking more questions, and if you have any questions, please tweet them on #MSQT, and we'll see if we can have time to ask and answer those things. So we've got a jam-packed session this morning because there's been lots going on in the last day. But first thing, I have to introduce you to my lovely panel. So on my right here, we have Dr. Ruth Dobson. She's a neurologist, so she'll introduce herself.
So yeah, I'm Ruth Dobson. I'm a neurology registrar working just outside London at the moment, and have recently done quite a lot of research with Gavin Giovannoni.
And here we also have Dr. Emma Gray. So Emma is the head of biomedical research at the MS Society in the UK. So she's joined us to take some and answer some questions, and that'd be great. And we've also got
I just waved.
Just wave. And actually, we've got another neurologist, a well-known face. This is Dr. Jeremy Chataway. He's a consultant neurologist at Queen's Square.
Yes, I'm a consultant neurologist and interested in trials and trial design. Here we all are. And we
maybe be able to take some questions from you online. But first of all, I guess what I'd like to do is really plow into Jeremy and ask him really what he was talking about today. Obviously, there's a lot of interest in clinical trials for people with MS, and Jeremy was talking about progressive trials. So Jeremy's got to pop off a bit later because he's a very busy guy and he's off to another meeting. But first of all, I guess, Jeremy, can you just tell us what you were talking about?
Yeah, no, thanks very much. So it was a pleasure to be here and talk about trials in progressive multiple sclerosis, which of course is the major problem. Looking back at the big late-stage phase 3 trials, looking at the trial design, and then how we must take the area forward. So, I mean, over the last 25 years, there's been about 8,000 people with progressive MS in trials. I think sometimes people think there's no trial activity in progressive MS, but there's been a lot. But it's been negative, as we know, unfortunately. All the immunosuppressive drugs, the beta interferons, didn't work in the way that we'd want in progressive MS. So people are starting to think about new things and think about new ways of protecting the brain and how to trial those in an efficient manner, how to look for drugs. I don't think there's a shortage of potential drugs, either from the laboratory or from other areas, other neurological areas. But it's how to get them through the trial process efficiently so that we can make sure that they do work and make sure there are no unusual side effects. So I'm particularly interested in what's called multiple-arm trials. So if a trial is running a horse race, then having more than 2 horses, the dummy drug and the real drug, having more than 2 horses in that horse race, perhaps 4 or 5 at any one go, and then we can see if we can get to the answer quicker.
Well, that's good. And you showed that there was lots of potential different trials ongoing, didn't you?
Yeah, I mean, it's an exciting time. I mean, over the next 2 or 3 years, there'll be at least 5 big late-phase trials in progressive MS, secondary and primary, starting off, I think, maybe in the autumn, I don't know, with the announcement, the top 9 announcement of the INFORMS trial. This is fingolimod, a drug that's used in relapsing remitting MS. We know a lot about it, but it's the end of the big trial in primary progressive MS with over 1,000 people across the world. So that's going to be very exciting. And there's more to come over the next 2 or 3 years. And then there's trials to take part in. For example, Bob Fox in the States, what's called the SPRINT MS trial, trying out a drug called Abudilast in primary and secondary multiple sclerosis. So look around for those trials and get involved if you've got progressive MS.
Yeah, I mean, one question that was asked to me yesterday by an MSer is, it seems that many of the trials are all about secondary progressive MS. What can you tell us about primary progressive MS? Because I guess many of those people feel a bit left out. And there are drugs for relapsing remitting MS. So what's happening?
So I mean, you're right. I mean, this is always a hot question as to how to do trials in progressive MS, whether to put people with primary and secondary together or split them out. I tend to split them out because I want to try and I want to increase the chance of success by removing what's called any variability. But that may be wrong, and many other people will put them together, and that's fine. But I think, as I said, there are trials in prime progressive MS. There's the Informs trial that will report, and then there's another trial of a drug called Mastinimib. So,
what's Mastinimib?
It works on a small molecule.
Oh, okay.
So plenty of things to do. Yeah.
And I think there was a thing I heard about that even more. There was this thing called the was it the Progressive Amala Alliance? So maybe Emma, can you actually tell me anything about that?
Yes. Hi. Yeah. There's been a kind of a really innovative and groundbreaking initiative that's been set up in the last year or so, and it's brought together for the first time in a really ambitious way all of the MS societies, well, most of the MS societies from across the world, and they've come together to put their kind of brain power and the money together to speed up developing treatments for progressive MS. And the aim is to spend about 22 million euros over until 2020. So it's a really large initiative. And what happened yesterday was the first 22 projects from the first grant round from that initiative were announced, and these were kind of innovative pilot projects. But what the Alliance wants to do is for the next round is to fund large international multidisciplinary collaborative network awards. So you're looking at kind of bringing top researchers from around the world together, at least in at least 3 countries, to kind of really build networks. So they want to look at finding treatments that can go into clinical trial biomarkers and improving trial design. So it's a really groundbreaking thing, and actually none of the societies have collaborated in that way before. So this is really, really exciting. So watch this space. There should be some information on the UK MS Society's website certainly about it, and the American MS Society's website.
Wow, so that's really, really encouraging, I would say. We've got some MSers online. Would you like to ask any questions? Maybe? Maybe not. Okay, well, if you do, jump in. So I've got a question that was asked by somebody, and it said, should CIS patients presenting with disabling symptoms and MRI and evidence of inflammation be considered for treatment with DMT? So maybe, Ruth, would you like to answer that one?
Okay, well, I think the first thing to say about that is that certainly those people with what we'd call high-risk CIS, people with CIS evidence of ongoing disease on MRI, for example, gadolinium-enhancing lesions, and also potentially with oligoclonal bands in the CSF, should certainly be considered for treatment. What was quite interesting this morning was the Barcelona group presented an update on they've got 1,000 CIS patients who they've been following up, and it's now for a mean of 7 years, looking at baseline values, so looking at what their MRI scan looks like when they first come in with their CIS, and also what their clinical assessment is and whether they have oligoclonal bands. And then also looking at variables at the end of 12 months, so repeating the scans at 12 months and seeing how well they've recovered from that initial relapse at 12 months to see can you actually predict patients who go on to develop clinically definite MS or who develop a degree of disability, so an EDSS of greater than 3, so fixed disability. And when they looked at sort of the 887 patients that had 1 year data and almost 8 years of follow-up, they had about 17% had relapsed in the first year, and the risk for second attack within this 8 years was quite strongly predicted by the baseline scan, so how many lesions were on the baseline scan, and whether there are any enhancing lesions on the scan, but also interestingly whether there were any new lesions at 12 months. So it's not just your baseline, but actually at 12 months that scan is also important. And when they looked at patients who'd had treatment versus none, and this was not an interventional trial, this was observational, so treatment was determined sort of outside of the trial, but treatment seemed to protect against that second attack, even within up to 8 years. Interestingly, oligoclonal bands didn't seem to predict the second attack, but what they did seem to predict is those who developed fixed disability within that 8 year point.
Can I ask Ruth a question? Can you explain to people what oligoclonal bands are?
Yeah. So oligoclonal bands are when you have when patients present either with MS or with a clinically isolated syndrome. A number of patients may undergo a lumbar puncture test. Not everybody will undergo this test, but a number of patients will. And what oligoclonal bands represent is evidence of inflammation within the central nervous system. So evidence of ongoing and active inflammation at that point. Once they're positive, they tend to remain positive. They're not within the diagnostic criteria of for MS other than primary progressive MS, but they strongly support the diagnosis in people with relapsing remitting disease.
So we've just got a question. I'm hoping to hear about a drug that switches off the immune system, talking about some research from Bristol. Well, actually, it hasn't been presented at ACTRIMS this week. It relates to a drug that will stop and dampen down the immune system. It was some encouraging work that was shown in animals about escalating the dose of a myelin peptide to dampen down the immune system specifically, and those clinical trials are ongoing. Actually, I think Jeremy might even know something about this.
Yes, thank you. I think I was the chief investigator. So, I think you're alluding to the Apatope ATX
I think so.
Trial. Yes. So this is work that does originate from Bristol, from David Rape and Neil Scolding, and they did an initial trial in about 7 or so patients with progressive multiple sclerosis, showed it was safe but no clear effect, and a dose-ranging study has just been completed in centres in the UK and Russia, looking at this technology, looking at it's given in the skin, either under or in the skin, and it's specifically in a way designed to tolerize the person against further attacks. So we took part in this study, and I was the lead investigator, and we haven't announced the results. I think the immunology has been announced, and that looks at an American meeting earlier in the year, and that looks encouraging.
So this is essentially like a drug for the future. It essentially aims to tackle just the bit that's causing MS without touching any of the other bits of the immune system, so the side effects should be less. But nevertheless, we've been hearing about some new drugs this week. Another one was a new variety of Copaxone, which rather than getting a daily injection, this is a 3 times a week injection, and that seems to work pretty well. And so obviously that's another way of moving forward. Now, we have had a tweet, and I'll just read it. It says, how long is long enough in studying primary progressive MS to know if therapy works, since it can be a slow progress? So.
Yeah. That's a very good question, because you really want to make sure that your treatment, particularly as a potent treatment, does work. And my personal view is, in the end stage, what's called phase 3 trials in progressive MS, you need 3 years. Yeah. Which is a high bar, but it's a necessary bar to make sure we don't go down any false alleys which ultimately would impede research in really making a difference. So I'm afraid it is a long haul. It's a marathon, but 3 years is required to really find the answer.
Okay. Okay. Well, that's a good one. So we have another question. This is, how many MRIs does a person with MS typically have in a lifetime?
I mean, maybe Ruth will come in, but I think that's interesting. In terms of diagnosis, they would probably have 1 or 2, sometimes to keep an eye on things if it's a bit uncertain in the early stage. Then the question, and it's a hot topic, as to how to monitor people more closely. Traditionally, we use clinical factors such as relapse rate and walking, but increasingly people are adding in MRI as a monitoring tool, and I think we're going to see a lot more of it, and it may be that a person requires an annual MRI.
Mm-hmm. So one of the ideas about drugs is really, treating it so the disease goes away, and this is called like no evidence of disease activity. Now, obviously, that's something you can only see if you look for it, and some of the outcomes have been MRI, looking for gadolinium-enhancing lesions. Another one is clinical activity, whether you have relapses or not, or whether your disability increases. But one of the new topics today was another potential outcome in terms of looking for disease activity, and they call this NEDA4, which was basically looking at brain shrinkage. And there was some work presented with fingolimod, looking over time and to see if there was a slowdown of brain shrinkage. And obviously, if the brain doesn't shrink, I mean, outside what normally happens as we age, because everybody's brain shrinking as we speak, and I'm probably losing lots of nerves right now. But, it was shown that fingolimod actually could really slow down this. So there was a quite significant number of people who had this NEDA4 activity. So again, it's basically how do we really ensure that actually disease is shut down, because that's one thing we want to do. And obviously, you know, annual scans is one way of doing it, but it obviously depends on where you are and what your neurologist does and their current practice.
I think Ruth has the details, because this is very interesting.
Yeah,
isn't it?
so this was this morning's presentation. Again, it was looking at the Freedoms trial, which was fingolimod in relapsing remitting MS, and going back and looking at the data and saying, well, we know that you can get patients' disease activity free if you use gadolinium enhancing lesions, relapses, and new T2 lesions as your criteria. But actually that looks very much at inflammatory activity in multiple sclerosis. So relapses and new gadolinium enhancing lesions are very, very inflammatory. And actually can we say, in terms of thinking about less inflammatory disease but atrophy, does fingolimod help with that? And trying to think about the rate of brain shrinkage that might be a useful target. So this is sort of a couple of figures, but 0.2% of brain shrinkage per year is thought to be what the normal population has, and that's higher in MS. And so, sort of taking a practical point of view, the decision was taken to use 0.4%, so slightly more than the normal population, but very, very close. And actually, about 20% of patients in the Freedoms trial on fingolimod fulfilled the criteria for new evidence of disease activity when you included brain shrinkage in that. And it certainly did independently contribute to outside of all of the inflammatory activity to disease freedom in fingolimod when you put it in. So it wasn't dependent on everything else, it was independent.
But I might also add that brain volume, I think, is the technique of the future. But it is difficult. It's technically difficult, and this is trial data. Everything is very carefully controlled. So we're not at the stage yet where you can go to your local MRI scanner and work out your brain volume and brain volume change, or the individual patient, because there's too much variability, too much noise in the system. But I think that's very much the direction of travel as we individualize treatments.
And I think you presented some data this morning showing that from the Lemtrada trial, where the brain shrinkage or brain volume can be kind of masked by things that may be occurring on. So you may get this thing called pseudoatrophy.
Yes, that's right. And in that trial, the brain, even though the person was on the active treatment, seemed to shrink a little bit more than it should do, and then it re-expanded back to its normal after 6 to 12 months. So it's not straightforward to measure brain volume. There are lots of factors, because of course what you're seeing is just the final result. But nonetheless, I think the community believe that it could be the most important thing, and we just need to work the technology onwards.
So it's a question of watch this space, I guess, which is obviously the mantra. I can see we've got somebody online. Would you like to ask a question?
Hiya, is that me?
Yes, it's you. It's very quiet though. Is there any volume?
I don't know if I can control the volume from here.
No. Okay, I can hear you. Just.
Oh, I can shout it. So I was wondering about PMR risk in natalizumab patient. Currently, my neurologist has mentioned to me that in their clinics they're finding that people that have positive antibody titer levels are actually fluctuating in time quite dramatically, so that their risk factors are changing every 6 months, which makes it quite hard to decide whether to stay on the drug. I was wondering if risk factors had been actually talked about in the conference so far, because I know there was, I'll need to look at my notes for this, is it L-selectin, using that as an additional biomarker on top of the JC antibody level to work out a much more accurate risk factor.
Okay, I don't know if we heard this. The question was the level of JC antibody titer can change and fluctuate, so that could potentially impinge on your risk of developing PML. And the question was, is there any work being done at trying to improve that? And it was mentioned that there was something called L-selectin, which may improve the risk or the thing of actually doing that. So I don't know if anybody saw anything on this. There's looking some blank faces, and I think there certainly was a session on PML, and but unfortunately I wasn't at that session. I think it was on when we were actually doing the Google Hangout, so I haven't worked out how to have myself in two places at once. But obviously Biogen are working very hard to try and work out how you assess the risk and how we mitigate that risk. And I guess things change every week, but I think what we do notice is that the number of cases of kind of PML are slowly coming down. So I think the risk strategy scheme is doing something. I don't know.
Yes, I mean, I think it's. I don't know the details, but it is work in progress, and a need to try and get a more stable index, an additional index is required. So we need to look through the details. I think it's not quite yet there as a clinical tool, is what I would say.
Yeah, I think that was. I wasn't at the talk, but from reading the abstract, it's early work and it looks promising that L-selectin may be an additional biomarker taken in the context of the anti-JCV titer, but the proof isn't there yet.
Yeah,
there is sort of very early data, so it really is a watch this space.
Thank you very much.
So sorry, I didn't give you the right answer, but obviously
there are many talks on it. It's fine.
Yeah. So have you got any other questions at the moment? Or should we just carry on, and then you can pop in as...
Yeah, I do, but I don't want to hog it, so carry on.
All right, we'll come back to you. You can always wave at me, and we'll stop. So, I mean, I guess one question is there are all these trials going on. How do people get involved in the trials? I guess I can put that through both to you guys, and also put it through to Emma. So
can we change the question to ask one for Jeremy?
Yeah.
Pop off.
Yeah.
Because somebody has a question on MRI that would be really good for Jeremy. Or
okay, which one?
This bottom one.
Okay. Different countries use MRI as markers to monitor progression. Some in the UK only use them when it's a requirement for the drug that a patient is being administered. Is there some sort of guideline that international community of neurologists can adhere to?
Okay, so I guess that's a bit of a build on the previous point. There is not a consensus at the moment. It's an evolving situation, and different countries do actually quite different things at the moment. So in America, for various reasons, there's lots and lots of MRI, and it can be done quite frequently, maybe 3 or 6 monthly. I've had patients with. Whereas in the UK, as we've heard, outside of diagnostic, it's done more sparingly. I think we will reach a middle ground, which we're not there yet, and maybe an international consortium, obviously ACTRIMS would be ideal, to maybe put the minimum of MRI, which may be annual, but we're not yet there with an international consensus, and different countries will be doing, different physicians will be doing different things as we evolve the area.
Yeah, I was going to say, even across the UK, different people have different approaches to how frequently they scan people. I think what has come out quite strongly, especially from this morning's talks, is that using MRI to monitor disease activity is potentially a very useful tool, and we should probably be doing it more than we are at the moment, but there is no consensus on that, and sort of on how frequently it should be to be useful.
Mm-hmm. Does that answer your question, Emma?
Yeah, that's brilliant, thank you.
Okay, I'm going to...
Well, thank you very much,
David.
Jeremy.
Bye to everyone. I'm sorry I've got to go, I really am. That was very nice. See you soon.
Okay, so I'm now the rose between the two. So, well, maybe I can ask Emma this question about, trials and how do people get involved?
Okay, so that's a really good question. And at the MS Society, you can imagine lots of people move closer. Lots of people ask us that, and there is actually there's quite a bit of information out there about getting involved in clinical trials. So if you want to look at the international trials out there, the best website is clinicaltrials.gov, and that has a list of all clinical trials, whether they're active, whether they're recruiting, information how to use, can speak to the trial manager. There's also a good new website in the UK called the UK Clinical Trials Gateway, and if you just Google UK CTG, that should come up, and it's got a really nice interactive map. But taking part in clinical trials is something that people should consider carefully, and you should always speak to your neurologist and healthcare professional, possibly members of the family. It can be a big time commitment. And there are actually lots of other ways that people can get involved in research as well as clinical trials. You know, there's the UK MS Register if you're based in the UK. There's
Said sign up.
Yeah, no, definitely. I think to speak up.
Oh, no.
No, definitely sign up. And I think probably Rod and Katie from the UK MS Register are actually probably in the audience watching. And there's the tissue bank. People can sign up to be a donor of the tissue bank. There's also other trials that don't involve drugs that people can get involved in. So there are sort of psychological therapies, exercise trials, fatigue trials. And there's lots of information if you wanted to go to the UK MS Society's website and you type in B in a study. We've got a whole list of studies people can sign up for. So there really is a lot going on and a lot of ways that people can get involved in research.
Okay, so we've had a few more questions. One question was for Ranjit about the MS Alliance, which I think Emma had answered. And one question was, are there new symptomatic treatments on the horizon? Have you seen any studies this week? I think most of the things I've been seeing have largely been around disease-modifying therapies, because there are some new ones, and there was a brand new one presented this morning, actually. So do you have anything to sort of tell us about that one, Ruth?
Yeah, so the one presented this morning was on a drug called daclizumab, which is a drug that's given subcutaneously monthly, and they did a trial of this drug, daclizumab, against interferon beta in patients with active relapsing remitting MS, so patients who had to have had 2 relapses in the previous 3 years, or 1 relapse in the year prior to study entry. And they recruited just over 1,800 patients, and who had a mean of actually 1.6 relapses in the past year. So these are patients who are having quite a lot of relapses, and they actually found a 50% reduction in annual relapses against interferon. So this is a drug that works really, and this is mirrored by the MRI endpoints. It's a similar reduction in the new MRI lesions, and again, it's important to remember this is all against interferon rather than against placebo. This is against sort of current first-line therapy. And interestingly, there was also an improvement in the other measures that they used, so things like a 9-hole peg test where you have to put pegs in the box, and also in a cognitive outcome. So it actually looks quite an exciting treatment.
I guess everybody would also say, what's the side effects?
Yeah, so it's always about what's the side effects, and the big thing that came out with this is that it's an injection people seem to get quite nasty cutaneous, so skin reactions. A number of people required steroid treatment. What they did say in the talk was that these are all reversible with steroid treatment or with discontinuation of the drugs. They're not a permanent effect. They are related to the drug, but they that was the significant side effect that was coming through.
So how often was the drug given?
So it's monthly. So it's just once a month injection. So big improvement on sort of interferons and Copaxone, which are obviously far more frequent than that.
Was it an infusion or?
It's subcutaneous. So it's under the skin. So similar. I think the idea would be in a similar manner to interferons, whereby it can actually be done by the patient themselves rather than having to come in, for example, like natalizumab, where you're required to go into hospital for essentially half a day a month. It could be something that could be done at home.
So
it's on the horizon. It's there, and it actually looks a very exciting drug.
So that's good. It's essentially more choice, which I think has got to be always a good thing. So that's quite exciting. Were there any other treatments that you'd seen?
So the other one that was talked about again this morning was generic Copaxone. So thinking about repurposing drugs, which has been quite a hot topic this year. And sort of alongside repurposing drugs is, can you get generic versions of drugs, so bringing down the cost to patients, to healthcare providers. And so the first of these really to come out is generic Copaxone. And they ran a study of it according to the European Medicines Agency regulations, and it comes out as exactly the same as Copaxone. It was nice to hear.
So competition,
Yeah.
and maybe a cheaper drug.
Good.
Let's hope so. I mean, Ruth just mentioned generics and repurposing, and there was an interesting session yesterday about repurposing. So what does that mean? It means it's taking a drug that you can use in one condition and then actually using it in MS. Now, you know, there's a huge interest in the government's funding these things, but in fact, it was presented yesterday that, you know, the drug companies are actually very good at repurposing drugs, and most MS drugs were actually invented for a different purpose. And so what it allows the drug companies to do is to get the drugs to you much, much quicker, rather than doing the whole development process. Now, as I say, most of these disease-modifying drugs are repurposed, but what I was very interested to see this morning presented was a case of repurposing really for repair. So again, working from academics, had been shown last year that there was a target for making immature myelinating cells make new myelin, and this was a histamine 3 receptor inhibitor. And actually what I also have heard is that this group are planning a clinical trial in the States with this very drug, but actually what I also saw this morning was pharma had been very, very quick off the mark, and they've taken a drug which was used in Alzheimer's disease, and they've repurposed it for MS, and actually already a clinical trial is already ongoing, and so far there's been 150 people recruited, and so that's really exciting, you know. So this information only really came out in November, and yet, you know, pharma have really been on board. They've got going, and you know, they're trying it. So, you know, these repair trials are happening. So that's really, really exciting as far as I could see.
Could I add something to that?
Yeah.
There's actually repurposing is a really, really interesting area, and there's actually lots of trials going on in sort of the academic research field as well. So there's a trial of phenytoin going on, and amiloride. One's based in London with Raj Kapoor and Matt Craner in Oxford, and they're looking at phenytoin and amiloride. And phenytoin was a drug for epilepsy, and amiloride for hypertension. So there's also Jeremy's trial that hopefully will start in the next few months of MS Smart, which will be looking at 3 drugs used for other conditions in an academic-led situation, and that, you know, speeds up the trial design, because then you don't have to do the safety phase again. You need to obviously find out if they're safe for people with MS, but we have some safety information, and hopefully the trials are cheaper to run. So I think repurposing is a really, really exciting area, and people with MS should be excited about that.
And I think you know, with these new trial designs that, you know, the academics are pioneering, is then pharma feeds into that, so they can then also get their drugs tested in these ways. And there is another study that's ongoing with a thing called anti-lingo, which is ongoing. So another trial which is specifically there to repair the myelin. So this is kind of different from some of the phenytoin trials, which are saving nerves.
And they're looking at slowing or stopping progression, is the idea. I should have said that.
Yeah, okay, so we've had another tweet, and this is an interesting one. Is there any evidence that sleep impacts progression? Since sleep may clean the brain, could it slow down the cascade of damage and inflammation? Whoa. So I'm going to put that one straight to Ruth.
I think at the moment, sort of directly answering the question, there's no definite evidence that sleep impacts on progression, but that doesn't mean it doesn't. It means that nobody's actually looked at it before. The research that's referenced about sleep cleaning the brain is relatively novel. It was only published a couple of months ago. So I think in the context of MS, nobody has really looked at that to date. What I would say is that there's certainly good evidence that symptoms may impact on sleep quality and sleep quantity, and actually, if that is being a problem, then you do need to speak to your physician about trying to improve the quality of your sleep, because that is going to have a huge impact on the quality of life, sort of outside of any evidence about progression.
I would say that fatigue would be a huge problem as well.
So we've had another question. Is there any news on the attitude towards patient-reported outcomes as a legitimate scientific tool? So I'm going to push this over to Emma to see if she's actually seen anything on patient-reported outcomes.
I haven't.
You haven't? Okay. Well, there was some studies, or there was a report of some stuff looking at iPhones and as a way of collecting information, but whether that actually makes it through into how a clinical trial is developed, I think is a bit for the future. But clearly, patient-reported outcomes are central and very, very important, and actually the core of many things. So, you know, as time goes by, these become more and more and more important. So,
I think they're becoming more and more investigated. I haven't seen anything here, but I know that it's a hot topic, and there are lots of groups out there who are desperately looking at validating them for clinical trials.
Yeah, and the one thing that I'd say is actually in the declizumab trial, there was a question about what the monthly drug was called, and it's called declizumab. They've asked me to spell it, and I've only got my own spelling written down, which is doubtless not correct. So I'm not going to give the spelling, but the name is declizumab. But they actually looked at one of their endpoints at actually a patient-reported outcome, so a fairly standard one, the multiple sclerosis impact score, the MSIS, and they actually found that the drug not only worked on MRI and EDSS, but actually had an impact on the number of patients reporting a worsening of their disease. So I think people are using them, and people are trying to think of ways in which they can be used better, for example, via iPhones, via online EDSS. But this again is sort of quite early and needs to be replicated again and again before it's rigorous enough to use in the clinical trial setting, I think, is a major issue.
So we've had another tweet. It said, has there been any talk about cost containment efforts to try and kind of harmonise the US towards Europe? Well, I don't know. I mean, I guess with drugs they tend to just get more and more expensive, which is obviously a problem. With the advent of the generics, of course, we hope that there will be some equity and maybe some price competition. But I'm not aware of stuff, but I'm sure it's a very important topic, you know, in terms of how you know, drugs cost in different countries of the world. Unfortunately, the Americans seem to subsidise the rest of the world in terms of the drug costs, because they're significantly more than they are in many other countries. But I can't really comment on that. Okay. What is the most exciting thing you've seen so far? Okay. Well, I can answer that one because I just told you. I think the exciting thing was this remyelination trial. You know, because I was so excited that it, you know, the information, the ideas have only just appeared, and yet pharma have acted on it really, really quickly. And you know, 150 people are already recruited into this trial. It's largely based in Europe. I noticed there was quite a few people in Eastern Europe, and there's 9 people from the UK. So I don't know much about it. So maybe if they're still recruiting, I don't know. I'll have to go and talk to the people when the posters are presented later on this afternoon. So, Emma, what?
I haven't actually got to see as many talks as I would have liked, so I can't say what's the most exciting thing, but I can say what an interesting talk I went to yesterday. There was a session on cell-based therapy, so they were looking at stem cells and how they work with the immune system and understanding more about how they function and how they could be used in MS. And there was an interesting talk from somebody that was explaining all the different measures you could use to measure changes in myelin if you were trying to repair myelin using stem cells. And she went through this list of all these amazing new techniques for measuring. There's one called myelin water and all these things, and it was just fascinating to see all these new tools being developed that, you know, sort of underground that we don't know about, to measure all these tiny molecules and proteins, and I thought it was absolutely fascinating.
I think there was some sessions again this morning on new kind of imaging modalities to, you know, work out how we assess repair and progression. So obviously that's a very hot topic as well. And you know, what we do need is good tools to work out how, you know, MS is developing and progressing, because obviously if we can get those trials much shorter, then that's what we've got to look for.
It's key, because you can do as much great research and have as many great ideas, but if you can't measure it and prove that it's effective, it's kind of ends up a bit of a waste of time. So sometimes it's a slightly overlooked area and can be a bit dull, but it's actually really, really important to have these biomarkers and these outcome measures and things like that.
And you heard Charlotte yesterday talking about some biomarkers that she was talking about. So I guess we put the question to Ruth now.
Okay. So I've had time to sort of think about this a little bit while you've been talking. It's a shame I didn't get to see the remyelination talk, because that really does sound very exciting. I think what's excited me this morning is the number of people that have sort of taken on board the no evidence of disease activity and trying to think more about how that idea can be progressed. So not just thinking about one talk that's been the most exciting, but the most exciting concept to come out, which is one that has been around for a while but hasn't had so much prominence in any ECTRIMS thus far, saying actually, you know, what do we need to be aiming for? We need to be aiming for freedom from disease. You know, a bit better is not really good enough, actually. No evidence of disease is really, really important. I think the take-up of that.
There's a bit more presented on alemtuzumab this morning, wasn't there?
There was the three-year follow-up of the alemtuzumab patients. So these is the two trials are called CARE-MS1 and CARE-MS2. So CARE-MS1 is patients who were essentially pretty newly diagnosed with MS, had no previous treatment, and CARE-MS2 was patients who had disease activity on treatments, patients typically on interferon who then relapsed. And both these trials were alemtuzumab versus interferon. But the three-year follow-up was looking at patients who'd received alemtuzumab throughout. And really what was exciting about this trial is that when they looked at year 3, so after two treatments, and then there was an option of patients, if they had relapses, to have a third treatment. Actually, at year 3, 87% of patients in CARE-MS1, so treatment-naive patients who got alemtuzumab at 3 years, 87% of them had no relapses, 90% had no new gadolinium-enhancing lesions, and actually 64%, so almost 2/3 of these patients were disease activity-free, so had no relapses and no MRI activity. So it's saying in 2/3 of patients that received 2 infusions at 3 years, they have no evidence of disease.
And that's that's kind of amazing, because essentially it's just a short sort of 2 courses.
Yeah, so it's 5 days in year 1 and 3 days in year 2. So yes, there are risks associated with the therapy, but this is something that in terms of the amount of time to actually receive the treatment is relatively low.
So on that note, there's a question. It said Lemtrada looks like a good DMD to manage relapsing MS, but short-term costs seem prohibitive if the outcomes are seen to be good in the long term. Would it not make sense to push for more of its use?
It's now licensed.
It's now licensed,
and the NHS have approved it.
So yeah, so I mean, I can only speak from a UK viewpoint. I'm a UK neurologist, but at the moment it has gone through NICE, and we are allowed to offer it actually as a first-line treatment now in the UK. So there is that push for its use amongst UK neurologists.
Yes.
I mean, it's also worth saying that even often when drugs are licensed and they are not NHS approved, people don't always get them, and it's a problem that I think the MS community is quite aware of and needs to kind of try and change, because it's not really good enough often.
Yeah. I mean, I guess it's NHS England are making their mind up, and I guess one of the things that Lemtrada has been licensed for is if MS is active. And somebody asked the question, how is active MS defined? So that's, I guess.
So how is that active MS defined? It's defined according to both your clinical presentation and your MRI scan. So it depends on your assessment by a neurologist. In terms of the difference in the services that people are offered around, and again this is from a UK perspective, around the country, I think with the increasing number of MS therapies that are coming online, the increasing complexity in actually choosing a treatment, it's more and more important that patients actually get to see a neurologist who's interested in MS, so an MS specialist, as early as possible in the disease.
Yeah, I mean, I guess that's one thing that's, again, coming out here, is that there's going to be more and more and more different treatment options, so which is good, but obviously with that, it's also getting the right information to know what each drug does and what its side effects are, and I guess it's where you get your good information from. So I'll give a plug for the MS societies.
Yeah, can I add to that? Yeah, I think that, David, that's a really, really good point. And actually, the MS Society have launched a campaign called Treat Me Right. If you want more information on that, it'll be on our website, because we're so aware of this problem, and the problem's several-fold. So there's kind of getting the money to pay for the drugs, there's information, there's changing the culture amongst neurologists, there's lots of issues there, and we're doing lots of campaigning to try and change that. So if you wanted to sign up to our campaign, find out information about all the drugs, so you're armed with the information when you go and see your neurologist, please go to our website.
Okay. So another tweet, patient compliance. Is anybody addressing how to keep us on course with treatments? Sometimes I feel I'm well enough to stop. Okay.
So it was interesting. Somebody asked. It wasn't a talk, but it was a question at the end of a talk this morning. Was you've done lots of trials about trying to step up treatment, and actually, should we have a trial about when to step down treatment? And the answer is we probably should, because nobody really knows, and especially with sort of more powerful treatments and potentially this idea of induction therapies, which are going to be, I think, talked about more this afternoon in the burning debate. But thinking about actually, can you ever step down treatment? But there's no information out there, and certainly from the natalizumab data, patients who stop the drug do tend to go on to have relapses on stopping the drug.
Yes.
And certainly we wouldn't be recommending stopping drugs without talking to a neurologist.
Yeah, I think that's the key, is talking to your neurologist about if you have any fears about the medicines you might be taking, side effects. It's always, I think, the best advice is to go and talk to your healthcare professional, and before you do anything rash. I think.
Yeah.
Can I add something about remyelination?
Yeah.
So I know you were talking excitedly about the talk you saw. There's actually I would say there's quite a lot of remyelination research going on in the UK, and the UK is kind of, a big pool of that. And there is a lot of research going on in Edinburgh and Cambridge, led by Robin Franklin and Charles French Compton, and they're doing loads of work looking at different remyelination targets and hoping to get those into clinical trial, and they're hoping to run a clinical trial later in the year. I know it's slightly further advanced, but I think it's really hopeful to know that there are loads of targets out there, and we hope that this pipeline is going to continue, and that you're going to see more and more remyelination clinical trials happening all the time.
Yeah, so it's showing that the MS societies do respond to what you're asking, and obviously the problem is time. Things don't translate as quick as you'd like, but nevertheless things are moving forward. If you think 5 years ago this wasn't even thought about, and now it's starting to become a reality. So very encouraging. And obviously, let's get our fingers crossed. But obviously, we should be cautious. The early studies, so we shouldn't be expecting the world. I know we want it, but things will evolve as they evolve. So let's keep our fingers crossed.
Can I add something further? Actually, often when a trial isn't as successful as we would like, you do often learn a lot of lessons for it about how to run better trials. So you can always get a positive out of any sort of bit of research that you do, and I think that's quite important to remember that even we do early trials of minor repair therapies, we'll learn a lot, and hopefully the next stage will be even better, and help give us better answers and closer to finding treatment.
Yeah, and I think that's one thing. I mean, we had the lamotrigine trial, which was seen as a failure, but in fact it probably necessarily wasn't.
No, we learn loads about how to run progression trials.
And what we found is some people weren't taking the drugs, and the ones that were taking the drugs seemed to do better. So that idea hasn't been killed off. In fact, the MS societies are funding 2 trials on different types against the same target. So let's be hopeful for those ones. Have we got any other questions? No.
We've done all our questions.
No, we've got more.
I think we've done most.
Can I ask you a question?
Oh yes, go ahead.
Hiya. It was just about it's following on from the definition of active when you define MS as active. In terms of defining it as progressive, there's kind of 2 parts to the question. The first is there a way that a neurologist can actually definitively know whether you're progressive? And also, does that mean that you are in the neurodegeneration stage and inflammation has stopped? It's all degeneration?
Well, I wouldn't say inflammation has stopped. It's very clear that in progressive MS, inflammation is ongoing. So I guess there are different types of inflammation. So there's the inflammation that tends to drive the relapsing remitting disease, and there's the inflammation that probably is involved in the progressive disease. So they're not quite the same, and they may need different types of treatment. So the question was, how do you define what is progressive? And I'll...
Okay. So thinking about defining progressive is often quite tricky, and it's quite tricky in the clinic as well. So what it relies on is repeatedly seeing the patient, so repeatedly seeing somebody and assessing whether there has been a change. So in the context of relapses, you see a clinical worsening, but then things improve again afterward. And the key for progression is that things don't improve, so that there is a sort of steady change or a steady worsening that doesn't get better. So that's how we define progression clinically. And just think, and to go on to the second part of the question, and David answered it partly about, does it mean that the inflammation has stopped? And the answer to that is certainly not. There's probably some degree of, well, there's certainly an overlap with pretty much everybody, I would say.
And isn't there some evidence, if I'm correct, about being inflammation in certain parts of the brain in progression, so potentially in the meninges, which is lining around the brain, and whether treatments like Tysabri is being trialled in secondary progressive MS, if that could be effective? We're waiting for the results.
We're waiting for the results of that one at the moment.
Well, actually, then that brings me to another bit of interesting information.
Excellent.
So, one of the studies that was presented today, I had a quick sneaky preview of what was on show, but it was basically looking at hot microglia. So, hot microglia is kind of an interesting target. It seems that maybe they're involved in progression, and what this study looked at was hot microglia in people on treatment with Tysabri. So,
Can I say what microglia are?
Yeah, microglia are like the macrophages of the brain. So, what's a macrophage? Okay, so what they do is they have lots of little processes, and they're
a cell.
They're a cell. Okay, they have lots of little processes, and they kind of feel the environment, and what they do is sense for change, and that may be, there's an infection here, come in and get rid of it. But it's thought that they may be part of the problem in MS, and the hope is maybe that if you control them so they're not so hot, that it may be useful for progression. And what this study showed, that was with Tysabri, although the activity of Tysabri is largely to stop white blood cells getting in the brain, if you kind of do that, then it seems that it dampens down the hot microglia. So by stopping the active inflammation, that it seems to control, what's actually going on in the brain, and maybe it's another useful biomarker that we can think of for, some new trials. And maybe it says, well, maybe something might happen in this trial. So we'll keep our fingers crossed on that. So that was another bit of things that I saw. Now, I guess we've kind of running out of time. So it just sort of really takes me on to, I guess, the last thing. The last question was, where is it? Can't find it.
Is it?
Oh, no, this is it. This is a question. It's like somebody asked us, watchful waiting, what's that? Does this include MRI, and should we be simply watching and waiting to see what happens, and then that decides how we should go? So I'm not actually going to ask Dr. Ruth the question, because what you need to do is you need to get online again this afternoon and join the burning debates, because we have two eminent neurologists who are going to debate the question, early and aggressive or softly. Early and aggressive is the only way to go. So have your say, join us on Twitter, join us, and take part. So I'd just like to thank my guests, Emma.
Hi.
And Ruth.
Hi.
For joining us. I'm sorry if we couldn't answer all your questions, but it's been a very enjoyable experience. We thank Alison also for organising and inventing this this new experience at ACTRIMS and ACTRIMS, and we'll look forward to seeing you again on a Google Hangout. So it's bye-bye from me, and it's bye-bye from them. Bye-bye.
Bye.